Hello everyone. So today I will tell you how we adapt a bioinformatic method to make it accessible for biologists by cooperating to develop the tool CellScanner. The first question is, why this project and for whom? I am a PhD student in bioinformatics. I studied biology for several years and start to learn informatics applied to biology in my master. So I'm working between the two fields. In our lab, we are working with microbial communities, and Anaïs and Charlotte are doing experimental research. They need to follow bacterial species amount in communities through the time. So to do that, they use flow cytometry to count the cells and 16S sequencing to have the ratio per species. 16S sequencing is a method which consists of extracting a specific DNA molecule, which works like a barcode for a specific species. This method is the most used in biology, but it has its drawbacks. 16S is expensive, time-consuming, and because it needs further experimental steps, it increases the risk of error. Anaïs and Charlotte can also wait weeks before having their results. But recently, machine learning methods have been applied to flow cytometry data to identify species. So we decided to use it in our cases. So first, what is flow cytometry? A flow cytometer is a machine where particles enter one by one through a narrow tube. When each cell is hit by a laser, the reflected, absorbed, or non-absorbed light will be analyzed by different channel detectors and converted into numeric values. These data indicate the size, the granularity, and the fluorescence of the bacteria. And we use this data to differentiate species with machine learning because we cannot do it by eye. Several articles deal with machine learning methods applied to flow cytometry data to identify bacterial cells. It is the case for this article from Peter Rubens and al. From 2017, which use random forests on synthetical communities. For this article from Bridge Dugan from 2020, they use neural network and describe a pipeline called CellScanner used on lake water samples. Other tools, such as Flock and Swift, are using clustering method to assess the diversity of a sample, but it doesn't allow the user to identify species. So these methods are really interesting, but the main problem is that there is no user interface, so you need to install several packages and the language depends on the pipeline. The function used need a lot of parameters that are not always explained, or their role could be unclear for biologists. And the output of this function are not or are simply displayed to the screen. So if the biologist needs it, it has to do supplement steps to save figures or table. So these methods need programming knowledge and they are not always accessible for biologists. After seeing these methods, we decided to use it, develop it, and make a tool which responds to biologists' needs, such as having a method description, having a comprehensive and easy to navigate user interface, but also having settings which allows the user to personalize the tool and accessible for them, and then produce useful results file and figure which can be used for interpretation. Then we defined the needs. We did it during the full tool development because this method was new for us, so we didn't know what we could expect from it. So when I was developing the first aspect of the tool, Charlotte and Anaïs were producing the experimental data to test it. From the test, I improved the function, and from the result obtained, Anaïs and Charlotte were defining more needs for the tool. As soon as an interface was in development, Anaïs and Charlotte was assigned as beta tester, and they applied their data to the tool to test it and formulate requests to me to solve the error and add new functions. The first need that we looked at was the machine learning method. So most supervised machine learning methods are divided into two steps. The first is the model training, which uses a monoculture file used in part to train a predictive model and then the rest to test the model. So we test with a small data set and judge the quality of it with this information. Then the model can be used to perform a prediction. The model performs a prediction from an unknown community file. But a small data set is not always representative of a biological experiment. So we added function called tool analysis, which allows the user to create an in silico community by mixing monoculture data. With this function, the program will assess the predictive model in the same way than during the training, but with other files and bigger dataset. With this function, the user can easily perform a complete analysis of the prediction capability of the program within specific conditions, specific species. This whole pipeline is called a run, and you can run it several times to improve the accuracy. Then we looked at the input. We implemented all the data preprocessing into the program. The user only needs to indicate the file names via the interface. The user selects directly one or several files from a file explorer, so it makes it easier. And we selected the default option for the best predictions. The output was really important for the biologist. It has to be easily accessible and has to contain all the potential needed data and some relevant figures. To save the result, we realized that having data file was necessary and put it in a specific directory, avoid file overwriting. So we create a directory each time a calculation started. The directory is named with the date and time information, and the format has been chosen to be ordered by date in the result directory, so results are easier to find for the biologist. The directory contains Excel files containing the training and prediction of tool analysis results, some figures, and a text file. We created this data file when we realized that the user didn't always keep track of the past experiments and the parameters used on these experiments. And for this program, the parameters can affect a lot the prediction. So we saved all the parameter and the function used in this text file. We can find the use function description, data linked to the prediction itself, the species expected, the values for the training and the values for the prediction, then the parameter linked to the flow cytometer used, then the advanced parameter used for the prediction. With that, the user can repeat the same experiment within the same condition. The Excel files produced vary from the type of function used, but we separated data in function of the step of production and of the importance for the user. So in a reference file, we have the statistics linked to the test done after the training. We have the accuracy and the standard deviation based on the different runs performed by the program. We can produce the same file for the tool analysis function since the program knows what result to expect. The prediction file contains the results of a prediction for one or several files, indicating the number of cells predicted by species, as you can see here. The learning stat file, existing also for the tool analysis steps, describe with more detail the quality of the prediction, including the true positive and true negative values, and statistical values such as specificity and sensitivity. We also produce these figures. The 3D graph compare the expected results to the predicted results. It is produced for the testing part, tool analysis function, and prediction function. The user can manipulate the 3D graph to obtain the best angle where the clouds are distinguishable and can also choose the axis. It also shows the accuracy when we know what to expect. Then the confusion matrix. It is really useful for the users. In case of test or tool analysis function, the user can compare the expected result to the predicted values. The color gave the information right away. If the diagonal is dark blue, then the program can distinguish species. Outside of the diagonal, we can find the false positive and false negative. Because sometimes users need the confusion matrix value, we added them in an Excel file with and without the normalization. Then the user can access it easily if needed. Another problem was posed early on the tool development, the gating. In biological sample used in flow cytometry, you can find cell and debris present in the medium. This debris is identified by the cytometer as well as cells, and we need to remove it before the calculation, before there are not cells. So there is not one method to do that. Each lab gets its own method, and one of the most applied is to identify visually the cluster which contains cells and use a lasso to keep only this information. This method is far from perfect. Nothing indicated that the selected event is all cells and that cells are not forgotten in the other clusters. Moreover, the lasso used in one case can be wrong for another sample, and most of the time, this kind of gating is based on a two-dimension graph. So what we have done is first we converted the lasso used in the lab into a mathematical function to perform what we call the line gating. But this function will also vary from one cytometer to another from the type of cell present in the medium. So too much variation was present in this method. So we developed the machine learning method. The program used sample containing only medium debris to identify debris in all sample containing cells. This method gets rid of the manual gating information, so it can work with all cytometer and all cell types as soon as the user furnishes the medium information. And as you can see, so here we have the same sample, but using two different methods, so the line gating and the machine gating. Um, the machine gating perform better than the line gating to identify cells. Here you have the two clusters, and here we can see that red cells are identified as are debris. The last need was defining some useful parameter for the user. As explained before, the program can perform the machine learning pipeline several times to improve the prediction. In fact, the user can choose the number of runs. For 10 runs, for example, the program will perform 10 predictions with 10 different models based on the same reference files. Then we develop what we call an average prediction from the 10 predictions. We count the number of occurrences per species for an event into these 10 predictions, and we relabel this event with the most prevalent species predicted. We do that for all the events and obtain the average prediction. The results obtained are improved compared to doing the mean of results cells per species at the end of 10 prediction, because we don't look global, but we look at each event. With these two former steps, we develop the unknown parameter. Because sometimes the species clouds are overlapping, it is hard for the program to decide which event remain to which species. And it results in high false positive value. So when we count the occurrence per species for each event at this moment, we can define a minimum of occurrence, for example, 70%. In this case, if the most prevalent species for an event is predicted more than 7 times, then the event will be labeled as this species. If not, the cell is labeled unknown. So with this information, the user can reduce the number of false positive, but the thing is, it will also reduce the true positive. So for the interface, we used PyQt5 with Python. Here is the main window. We decided to locate the function according to the user needs. Button for the main function prediction and tool analysis are here. These are the principal buttons, so we have to access it quite easily. Then we have access to the reference data. The user needs to complete this information before doing a prediction. But once selected, this can be used for several predictions, so it doesn't need to be used as often as a prediction function. We put on the taskbar the advanced parameter, which doesn't need to be changed once it's defined. The parameter match the expectation. So once or sometime, but really less often than the other button. And because we realized that one cytometer to another, the channel's name were different, and because not all of the channel's information is useful, we created the cytometer parameter button. The user can define the cytometer channel name by importing a data file from this cytometer and choose which of these channels need to be used for the prediction. Because the biologists used sometimes the same species and same reference file, we decided to save this information in a database. We can find on this data relative to a cytometer, including the channel names to consider. The data linked to the references, the user can create classes which contain records such as species like E. coli, and reference files such as a specific monoculture are linked to each record. And finally, all the advanced parameters are also saved in the database, as well as default value, and then can be restored by the user by clicking a button. To avoid basic error, we reduce typing. So for example, here, all the inputs can only be integer or float. We have checkbox, and all the things that can be selected have to be selected from a list. We created a button for every step and implement error message in case of incompatible data, missing information, and ask for validation when creating or deleting an entry on the database. So you have here an overview of the program open on the update data function. So the user can create this space entirely by adding new entry, and you will have this window, and can add file by adding a reference and looking for this file with a file browser. Now the tool is done, we decided to distribute it. So the tool is compatible with Windows, Mac, and Linux. We simplified the installation for Windows. No dependency is needed. So we used PyInstaller and NSES to create an installer, which also creates a shortcut for easier use. For Mac and Linux, the user needs to install Python and some packages, such as pyqt5, but we reduce the installation at 6 command lines. The package can also be cloned from the GitHub repository, and function can also be run with command line for advanced user. All these versions of the tool can be found on our lab page, so you can follow this link. Finally, we are writing a user manual explaining all the function, method, and parameters. Some tests and tutorial will also be added for the user to train on the tool. A shortcut is created to access the help file more easily. The development of the tool may be imperfect, and we change it through the whole process, but the tool is now usable by biologists, and it is already used in our lab by our teams. And it doesn't need programming knowledge, so the user can adapt it to their own needs thanks to the database and have a quick check in small communities composition. So I would like to thank my lab and all people working in this project, the organizer of this conference, and you for your attention. Thank you.
Thank you very, very much, Clémence. Okay. We have a few questions. Just wait a moment while we load up. Looks like we can, here we go.
Yeah, okay.
Hi, Clémence.
Hi.
Thank you very much for the talk.
Thank you.
So we have a few questions. The first one is about the what did you use for auto completion? Was it a module, or how did you do that?
For the auto completion, I use a function from PyQt5, was included in the package. which allows you to complete the word from a list of words already existing. So I put a list of all the bacteria in the database, not exhaustive, but I try to put the maximum. And the user can auto-complete the species when it goes on the database.
Another question, do you know if the tool is being used outside of your lab already?
So I know that some person already downloaded the tool and use it under Linux. So I don't know if they use it actually right now, but it's downloaded already. And I'm trying to solve the problem that we could have from other version of Linux and Mac, for example.
Out of my own curiosity, how are you tracking them?
Through my GitHub webpage, because for Linux and Mac, you need to download the package, or you can clone the GitHub package. So if you have error message, you can also ask question on my GitHub page, which is available. You can go to the page by following the link, because of the web page, there is all the link to access it.
Okay. Maybe we can put that link in the chat, or maybe you can put that link in the chat a little bit later.
Yeah.
We have another question. I'll have to read it verbatim because it's quite long. I guess you are continuing to develop new features and bug fixes for your software.
Yeah.
How are you convincing your users to update their environment when there are new versions, in particular when you're fixing bugs? Yeah, that's the essence of the question.
Yeah, so for that, I don't have an answer yet, especially for the Windows version, because there is the installer, so you will need to remove the old version and reinstall it, which can be annoying, especially if I do some modification on the database, which cannot be saved from a version to another. So I didn't find a way yet to change that. So with, yeah, I have to solve this problem and find a way. So we create a version of CellScanner. So we are at the first version, but yeah, we don't know yet how to contact or ask people to download the new version to do that.
There's a comment from Bjorn who that might be suggesting a solution for this. Is he says, to avoid the installation process, do you think the modules could be integrated into Galaxy or other workflow systems?
It may be possible. So I didn't look at it yet at all. Just wanted to, yeah, put an installer or something really easy for biologists. But yeah, it could be a solution to have the interface and remove all the installation problem. Yeah.
Okay, the next question from Alex prompts something for me as well. He says that maybe Bioconda would be an option. I'm thinking that the Python package index would be another place to be able to distribute it.
Yeah, so the only thing that if I want to do that, the only thing that I have to be careful is see if the user interface will interact in the same way via Bioconda, because I know that sometimes there is a compatibility problem, but it could also be an option, yes.
Okay. Another question. When you said something about using a model, is that model something that is configurable? Do users select that, or how's that?
So there is different so predictive model that can be selected. So there is different methods: random forest, logistic regression, neural network. So we put the 3 different functions into the program because the running time of the 3 different programs are really different. And for some bacteria, which are really distinguishable from each other, for example, logistic regression would be enough to distinguish them. So we gave this possibility to the user to select which method it can use. And of course, they can select some parameter into that, like the number of cells to use to train the model and to test it. And the ratio, for example, of the testing part and training part. But some other parameter, we removed it from the hand of the user, for example, the solver used or the number of layer for the neural network. because we took the decision that it was not the parameter that the user will use the most. So we try to find the best one, apply to these functions, and keep them on the Python function that are not accessible for the user interface. But for the one who use the function directly without using the user interface, if you download the package directly, he can access these parameters.
Do you foresee being able to infer those parameters rather than having them fixed? The ones that aren't user configurable?
This will also depend on this one, the configurable parameter. So there is a default value, but it will also depend on the input data. For example, the size of the file that you gave in tree can allow the user to train the model with more cells or less if the file is bigger or not. So it can change this parameter according to that, for example.
Okay. Slightly different topic. When you're beta testing with users, were they giving you useful feedback always, which is hard to believe, and as it's likely not the case is that they're not. Were you coaching them in how to give you useful feedback?
So in the feedback, I will say that there is the easy feedbacks that are just about the interface or something to facilitate the lives of the biologists, like putting the results in alphabetic order or putting the directory name in the certain configuration to have them ordered by time or date. So it was quite easy to fix and not affecting too much the program and the use of it. But thanks to the biologists and how they work with it, because they work with it through all the process, we were able to identify some major problem. For example, the case that the flow cytometer are changing a lot from a lab to another, and that the parameters are way different. So without them, I will not have been able to identify this kind of problem. So yeah, they gave me quite useful feedback for that.
Okay, great. Thank you very much.